Witamy po krótkiej przerwie. Teraz wzbogaceni o wiedzę z wykładu Katrin będziemy chcieli podyskutować o problemach neuronauk, tych bezpośrednio dotyczących mapowania mózgu, ale także szerszych, bardziej ogólnych i może nawet filozoficznych. Jeżeli Państwo mają jakieś pytania i chcieli je przenieść właśnie z wykładu na teraz, to bardzo proszę się zgłaszać. Ja troszeczkę słabo widzę przez te reflektory, ale proszę podnosić rękę i będziemy je włączali w tę dyskusję. So, let me start. You actually touched upon all the interesting topics in your talk. There isn't much left to discuss, I would say, but we can always try to develop the ideas that you mentioned. And you mentioned it several times that you would like to achieve a map of a multidimensional brain, so not just static map, not the universal brain, but also to develop it in other directions. So there are two directions that we are all interested in. One of them is aging. So can you tell us about the work and about the, if not ongoing work, but then maybe about conceptual work on mapping the brain as it ages, so as it develops and ages, and what do we know about it, and where do we, what are we aiming for?
Well, the brains that we have used for our atlas are brains coming from body donors, and their average age is about 63 years. So these are adult brains, and we are studying 10 brains, so that does not really tell you about the trajectory of aging. And there's a lot of variability in individual aging. So I would say to understand aging, the better methods of choice would be MR imaging and to study large cohorts like in the UK Biobank, or there are other large cohorts. So in order really to identify the trajectories of brain aging, it's necessary to have large sample sizes, and this is not possible in postmortem brains. But when you have identified in these large cohorts spots in the brain, then of course you can go into histological studies or genetic studies or molecular studies. So the concept would be for aging starting with in vivo imaging.
So the question is, of course, you can imagine that the cytoarchitecture of the 63-year-old brains might be different from than the cytoarchitecture of a 30 or 20-year-old brain. Do you actually have problems acquiring donors? How does it look from the practical standpoint? Do people actually think about it? Do people contact you and say, okay, I want my brain to be in case, well, I know it sounds very, very brutal, but that's how it works, right? Do people actually think about it, or do you reach out to people, or how does it work from the practical perspective?
Well, the brains are coming all from body donors, and they are contacting the anatomy department or pathology department of the university. And then there's a very clearly defined process and an ethics committee and so on and so forth. And we are getting the brains then for processing. But we cannot process so many brains, so maybe one per year, yeah, because it's extremely time consuming to cut the brain into so many sections. So we are not very fast. And I would say, no, there is not such a big problem. There are many people ready to donate their brain to help education of students, or also when they feel that they had a disease and they would like to help that others then better understand the pathomechanism of the disease. So there are many people aware of body or brain donation, and I would say that's good. My personal impression is these people are getting very old, 80 and 90 years, so maybe that's a good recipe. When you donate your brain, you are becoming very, very old.
Oh, yeah. Or if you are so aware of the necessity or of the higher cause, that means you are a person who is more likely to remain healthy and old, because
who knows? I
mean,
mean,
aware of those issues.
That's true.
So do you have to compete with the plastinarium, or they are a completely different market shelf?
That's a market, and anatomy is, so to say, these are really people who just want to to help in certain way and help education, help teaching and science, I would say.
And so aging is one dimension. So this also answers to some extent the question that the students just had during the break. Like, so the question is, how far are you in your research? And it's just impossible to define, because whenever you complete one project or you feel one assignment, then another one shows up, right? So clearly, aging is one of the dimensions, but another one that you just mentioned is the neurodegenerative disorders and generally the disease. So are there any projects that are parallel to the or that are aiming at mapping the brain in disease, and how are you able to incorporate that data and put it on the map?
Yeah, with a map we can help to better identify brain regions. And we know from early pathological studies of Heiko Braak, for example, that Parkinson has a certain staging or Alzheimer's disease has a certain staging. And using the maps, we can see these effects in living human subjects, in living cohorts. And it helps really to better identify those brain regions that are more susceptible to these changes during aging. So Atlas is a tool to better localize findings, I would say. And at the beginning of neurodegenerative diseases, it is sometimes very difficult to distinguish the different types of diseases. Yeah? So it's not so clear whether it's a Parkinson or another disease. And when following up the course of the disease and imaging patients from time to time, we can try to identify patterns that are then typical for a certain disease and also help to better develop diagnosis, or also to better understand what is the dynamic of a certain pathological process. And for patients and their relatives, it is important to understand how fast the disease is progressing and what are the perspective, what is the prognosis. So there are many possibilities of using the atlas for quantifying changes also in the living human brain, and this is what we are actually doing.
We also discussed it during the break. Is it easy, or is it becoming more easy to incorporate the data from animal models? You mentioned, and we saw it, it was beautifully visualized how different the rat brain is from the human brain, but we could also see some parallels and some similarities. So do you think we're with the development of our techniques, is it more difficult to incorporate the animal model data, or it becomes easier to make the parallels, to draw the parallels?
I think it's becoming easier because we can use technologies like polarized light imaging or cell body staining or immunohistology and do it in different species and then try to find out what are the similarities and what are the differences. And we should look very specifically to different brain regions, and some regions are rather similar between the different species, and we find even similarities between the pigeon brain and the human brain, and everybody thought they are so far away, they branched in evolution such a long time ago, but now we see that in certain nuclei of these pigeons, they have a very similar structure as human brains have in the cerebral cortex, and this speaks about the functionality also. So I feel these comparative studies are extremely important and relevant.
Yeah, so I would continue, and also maybe maybe I would want to give some more information to the audience. The animal models, of course, are not as precise in terms of neuroanatomy, but the animal models allow us to use certain tools that are unavailable for the human research, right? We have transgenic animals that have fluorescent neurons, so you can trace them. You can have a full map of an entire brain of a mouse without slicing it, without cutting it, and you see individual neurons in action. You can see them where they project. You don't have to slice it. So obviously, studying smaller brain of the model animals is expanding the range of applications available to us, right? But on the other hand, we're still limited by this difference in anatomy and the complexity of the model brains. So it is really important, it is really critical to be able to connect these two worlds and to make parallels. So for example, the polarized light microscopy is really fantastic because it does give, it could be compared to other tracing methods used in rodents, and then you can draw parallels, and you can draw conclusions directly from the animal models, right?
Yeah, and I think, I mean, what we tried in the HPP and what we also succeeded, we have built an atlas that consists of a human brain atlas, of a monkey atlas, a rat atlas, and a mouse atlas. And the same technology is used, and also the names of the structures, we call it ontology, is comparable, so that we can really start now to compare brains between the different species. That's also a progress, because usually you have only a rat brain atlas or only a human brain atlas, but when we want to do comparative studies, we also need to make the atlases comparable, and this is one of the achievements that we had in the HPP.
You can draw really unexpected conclusions from evolutionary neuroanatomy, because sometimes you see how particular structures change relative size or relative positions, and you can infer from that how the brain function changed, right? How the contribution, for example, the input to hippocampus, the medial and lateral entorhinal cortex have changed their proportions during evolution. That means that the spatial component of the hippocampus is decreasing and the non-spatial component is increasing. So in our hippocampi, instead of just tackling the spatial memory, they started to tackle other aspects. Like human, of course, human brain has different requirements than a rat brain. We think of and categorize and remember many more things than a rat. That is reflected in the changes of the inputs from these two structures to hippocampus. So having a full repertoire of or full gallery of species is also very informative to us, right?
Okay, let me also ask a few questions. So when you have a mega project like the Human Brain Project, the obvious comparison, of course, is to the Human Genome Project. The Human Genome Project has delivered amazing things, like concepts like the microbiome, or the concept, we sequenced the Neanderthal. I know now who are my ancestors because I tested myself in a commercial service, 23andMe. And I think it happened because of advances in technology. Like it suddenly became 1 million times cheaper to sequence. You could sequence all the bacteria in your gut. You could sequence little pieces of bone from the Neanderthal. Now, do you think we can have another breakthrough in brain mapping? Like, okay, we have some progress in scanning, some progress in optogenetics, but what I dream of is a technology that I would see every neuron, single neuron level, real time. Okay, so am I asking for, equivalent of teleportation, something completely impossible, or do you think if we invest right, there's a potential for another breakthrough in technology that will give you something like the Human Genome Project has given to the society?
I think so, yes. When we have the Human Brain Atlas today, it's an atlas that is going from the system scale, from the whole brain organ, down to the cells. And we have 86 billion nerve cells. And we are now capable to image also all these cells, and we are working on a 3D reconstruction also at the cellular level. And this is simply a new dimension of research that is possible, because we now can really better understand not only the single neuron level, but how the cells are being connected to each other. And I think when we really want to understand brain function, we should not only look to the single neuron level or also to the gene expression at the single neuron, but we should better understand how they form networks, how they form layers, brain areas, how they interact with each other. And therefore, the technological foundation is now laid. We needed computing for it because the storage and computational requirements are very high. We needed a certain level of neuroscience, and this is also now given. But now it's open, so to say, the door is open to really understand how this most complex networks is interacting, at least on a structural level.
So you think that with the current technology, with just the right amount of mapping and pooling and modeling, we can understand that let's say, for me, the problem is the resolution of the in vivo methods in human. I see, when I look at the brain, I see millions of neurons, what they do. I cannot see what they're doing together. I cannot see the computations they're doing. You think the current technology, we can break it? There's no need for a new method that would radically transform what we can do? Or no possibility for one?
Well, there are different approaches in the world approaching, I would say, different topics. So the Allen Brain Institute or the Brain Initiative in the US is approaching the single-cell genomics level, and they are very systematically analyzing each and every single cell. So we were more focusing on the systemic level, so how cells are interacting, what's their architecture, and what this has to do with the function. And these worlds have to be brought together at the end. And we are still far away, I would say, from bridging the gaps between the molecular cell to the systemic cell. This is not yet happening. We have it only for certain spots, sweet spots. And we are also a little bit away from understanding how the human brain, how it's changing. And we spoke about aging, but we should also speak about development, ontogeny, plasticity, the way how we are learning. And this has to do something with human-specific features that we do not really well understand. And I would wish really we would have more breakthroughs in much more brain diseases. Digital transformation brought progress in certain approaches like measuring consciousness, like helping patients with paralysis to walk again, or like epilepsy surgery. But these are only 3 examples of many, many that we cannot yet tackle. So for the next year in medicine, I see really the need to understand the whole pathogenesis, but not on a, so to say, on a level only of cells or only of molecules or only of imaging, but really in a more comprehensive way.
Yeah.
And this is only the beginning now.
Okay, one more, actually. So we're talking about the artificial intelligence and the neuromorphic possibilities, and how somehow there's a danger of this technologies ending up in the hands of just few American companies. Well, from a perspective of someone who studies the human brain and how it's influenced by air pollution or socioeconomic factors. I mean, there's plenty of data that is actually produced by digital platforms like Google or Facebook. And having this data would give us amazing tools to understand human society interactions. But I cannot access this data, even if my colleagues in the Max Planck Institute or the Jülich cannot access this data. You need to be in Stanford or MIT and have very good relations, personal relations with the bosses of these companies. Do you think it's possible for us, for researchers, to get somehow access to this? I mean, this is a much bigger picture, because what you really need is either to transform how we treat these platforms, maybe treat them as a public utility, or to have European platforms. But the best thing, of course, would be to work with our American friends. but being close to circles where I assume where these things are debated, do you see possibility that one day this data will serve research and let's say the public good?
I think this would be the right thing, yeah, to share the data. I mean, the data are coming from people like you and me.
Coming from us.
It's from us, yeah, and we should have access. But we also know that economics sometimes do not work like this. So I'm convinced that Europe should have own databases and own algorithms and own approaches to generate the data. And I mean, Google and Amazon, they have, of course, huge amount of data, but we also have very high quality data. Research data is high quality data. It's well annotated, it's reproducible, and it is not dirty, so to say. So it's really high quality, and we should make sure that that we use it and further develop it. And the Human Brain Project made some effort in order to overcome fragmentation also in the generation of data and production of data and really focus it, integrate it so that then everybody can use it. And in my view, fair data is one of the guiding principles that we should follow, and we should not necessarily wait. For our American friends, we should collaborate where this is possible, but the example that I showed with Lion illustrated that also with rather small effort but nice thoughts and creativity and engagement, we can reach steps that are comparable, and then we are competitive, and then we are also in a better situation to discuss with our partners whether we can exchange or share.
Yeah, there was a question that is somewhat relevant to what we just discussed. It might be a little bit specialized, but you mentioned the paper that was published in an open source that everybody can contribute to. Do you think this way of reporting of scientific data might take over in the future? So there's a huge crisis currently, even in the open publishing. There's already a system that started to milk scientists and abuse the system. Do you think this way of publishing with contributions that's not based on journals or just based on peer acceptance and review and contribution, do you think it might take over scientific publishing in the future? Or this is just an isolated example?
I do not think that's an isolated example. There are several examples like this, and bioRxiv or Zenodo are platforms that are being used. Peer review has its value and its merit, of course, if really colleagues are seriously reviewing papers and trying to make them better and bring them to a higher level. And there are several journals which have a very high level peer review system. And as long as there are people there who make sure that the system is there, that's fine. Other journals are more in a crisis. So Neuroimage had quite some problems with the editorial board just recently. And I think it's justified. We should make sure as scientists that the journals are not only seeing their economic values and neglect, so to say, the scientific and other values. And that will be a hard fight. Of course, in today's time, but I think specialized peer-reviewed journals, they are extremely important because they give the necessary feedback, and independent control of what has been published really has a certain level of quality. I think there's a second development which is extremely important, and that is sharing really the data together with the manuscript. Not only have the manuscript published, but also have the original or some process data published so that everybody can recapitulate and reproduce the findings, and then the data and the findings become better and have a higher quality. And that was also an extremely important step of many journals requiring that the data are being available together with the text.
Excellent, thank you very much. Are there any questions from the audience at this point? Oh yeah, there's one. There's two. Okay, so let me just get you. Sorry.
I would like to ask whether in your project, do you divide brains into female and male ones? Because many animal brain atlases, such as Paxinos Atlas, they are based only on male models, and they are very often used in neuroscience in rat atlases. And another thing is that some recent modern studies, they start to abolish the sex differences. And so the question is whether you would like to divide and create two different sex atlas.
Okay, the atlas that I have shown you for, which is based on the cellular architecture, we are always studying 5 male and 5 female brains. This is a small sample size, obviously, but we always make sure that we have both sexes included. And I think that any experimental study should consider both sexes, obviously. And I mean, there are even other factors when you think about estrus, diestrus, so there are even more factors that are influencing brain structure and brain function. We have, if you would like to better understand what are the structural differences at the level of the brain for behavior, it's becoming much more difficult, obviously. And there are many dimensions that are influencing gender, for example, and there is no clear relationship between sex of the brain and gender with all its dimensions. And these relationships are not well understood. And as somebody coming from postmortem studies where we study only whole brains, it's very difficult to understand or to tackle this problem because we simply do not have enough brains to answer this question. But to do research regarding sex differences is extremely important in human brain research. And there are so many examples that drugs have been developed for patients, for example, that were tested only in male subjects, not in female, but female subjects reacted in a different way. So there is certainly a necessity to explicitly address sex differences and gender differences in neuroscience. And in the Human Brain Project, I mean, at least we did some attempts to promote this type of research, and we had little awards in order to encourage students and researchers to do this type of research. But it's not yet well done. It's only at the beginning, I would say.
We have another question from the audience.
Yes, something probably a little bit aside to the topic, but when we talk about neuroplasticity, usually we talk about how fast are we learning certain things or certain skills. Is there a difference to this plasticity when it comes to unlearning things? Is that even like studied, how if the brain is able to detour from the patterns that it used to follow?
I would assume that this is the case, but I did not study it. But I know that it's one of the most exciting topics. And when you transfer it to computers and technology, I mean, this is one of the major differences that we humans have some also ability to dislearn, and technologically that's not yet there.
And sorry, another one, do you think that there is a capacity that the brain has, like a certain capacity to, like how I imagine this, because I'm not in the field, like there is a certain storage, right, of the memory that the brain can have, and there is this processing capacity. And given today's circumstances that we are so much exposed to the amount of data in the world, we probably would start to lose this capacity of the brain to function properly. Is that studied in some ways of the models?
Well, it's an exciting topic, but it's really, well, it's a little bit beyond my own research field, and I do not want to speculate here on this.
Thank you.
Let me just make a quick comment about the unlearning and forgetting. So sometimes we forget that brain is not a computer. It's an organ that is supposed to perform certain functions, and erasing is very rarely, actually it does not occur in brain, or very rarely occurs. Unlearning is also a form of learning. So when we overwrite the old memories, we create new ones, but it's not, you cannot just hit the delete button and move it to trash or erase it physically. So this is always a process. So memory consolidation, reconsolidation, forgetting, and memory erasure. These are different processes. So it's much more complex than in the case of a computer, where we can actually force it to delete something or force it to forget it. In case of the brain, I mean, unless we do it in a very brutal manner, because sometimes we have the case of amnesia, but that's not really physiological, right? So physiologically, it's not, these processes are not as binary. They're always a process, right? And we have another question here.
Yeah, thanks. In a way, this question is, I think, a little bit related to the comment you just made, but you used the word circuit in describing certain things happening in the brain. And in a way, that's a metaphor for the brain being like a computer. And I'm wondering if there's ever a danger in using that metaphor in limiting the way we think of it and sort of forgetting that it's a metaphor. Or maybe you don't consider it a metaphor. Maybe you do think of it as a circuit like a machine. So that's one question. And then more specifically, near the beginning of your talk, you gave this the number of 100 billion neurons and how many synapses, and then you said each of those can have two putative states. And that creates kind of an image of it being sort of like a binary computer, the two states. And so my question is it always a binary? Are neurons like on or off, or are there possibilities where there are other states and it's not just one or two?
Well, the neurons are firing or they are not firing, but the neurons are always part of networks, and this makes it so much more complex, and there are different ways to modulate, and even the synapse is a little universe where you have different receptor types that are acting. So it's even more, on the one hand, it's even more difficult because you have more possible states. On the other hand, of course, not all theoretically possible states are realized in nature. So in that respect, it's a metaphor, but it's nice to show, in my view, the complexity with which we are dealing. That was the only thing.
We have another question from the back of the audience.
Thank you. I wanted to ask, maybe it's a difficult question, but does the Human Brain Project help in any way, is useful in any way to solve the problem of consciousness? What consciousness is, how does it work, and where does it come from? A few years ago I read a research of Professor Christoph Koch about claustrus structure, that claustrum, yeah, claustrum, przedmurze, po polsku przedmurze. No, doesn't matter. That maybe it's there. So does Human Brain Project say something about that problem?
Okay, Professor Koch is not defending the claustrum as a seat of consciousness anymore. I mean, that would be, I mean, he came to more differentiated views of consciousness. Indeed, there are different theories trying to explain consciousness. There's also the global workspace theory, there's a Stan de Haan, there is Tononi. What we tried in the Human Brain Project to take such approaches and collaborated with people from Milano, Marcello Massimini and Stephen Lores from Belgium. And these are two doctors which are using, I would say, Tononi's approach in order to measure consciousness in patients with coma. And they can estimate a certain number that tells physicians how deep the coma is in patients with a loss of consciousness. And apart from the philosophical question and from the basic science question, this is of course a very interesting approach, because knowing what the level of coma is extremely important for doctors, but also for relatives of the patients. And it opens also a possibility to develop wake-up procedures for the brain, if this is possible. So the research on consciousness that we did in the HBP was focusing on a little and specific question, but it has consequences to medicine and hopefully will further push this research. But what is consciousness still remains one of the mysteries that we would like to understand. But handling it in the way that we can make a benefit for patients, this has already started.
So can you say that actually chasing the big question has generated technologies and methods to help comatose patients?
Yes, I think this is one of the outcomes of the Human Brain Project, which is of course not only our merit, obviously not. I mean, the colleagues started, they have other collaborations, but we pushed it and helped to develop it, yes.
I think we have reached that stage of our discussion that I predicted. So we have ventured into the philosophical questions, and this is the moment where I feel obliged to ask the question that appeared on the chat during your talk, which was: is there such thing as free will? And what do neuroscientists think about free will in the light of experiments that show that the brain activity predicts the individual or the individual feeling or the individual perception of causality? So do we think as a community, or do we think as experts, do you think as the ultimate expert that such thing as free will is, or is it just some kind of meta phenomenon?
Well, the question of free will was very hot, I would say, and there were the philosophers on the one hand, there were certain colleagues from neuroscience also at the other hand. And I feel before you, so to say, it's very important to define very precisely what do you understand as free will. And when you refer to free will as a type of behavior in the society that has legal consequences, for example, yeah, as human beings in a society, do we have a free will? Yes, I think we should accept this on a societal level. From a level of neuroscience, this question is much more difficult and controversial. Certainly the first experiments, Libet experiments, in my view, they were not suited to answer this question. They were not, I would say, good enough, and still also for subsequent experiments, there's a lot of criticism to answer these so to say, this question. It is highly difficult to operationalize this question in terms of brain science and of processes in the brain. And this we see in many occasions, I would say, when we come from psychology, from our daily understanding, what is language, what is attention, what is social behavior, then this is not necessarily what we see in the brain in terms of a physiological activity. So these two words are not yet connected, and this makes also the free will question a little bit ill-posed, I would say, for neuroscience.
Yeah, definitely. It's important to operate the same language and give the same meanings to the terms that we use. So sometimes that causes the confusion, right? We thought about it in a slightly different manner. I think we're approaching the 40-minute mark that was imposed on us by the organizers. It's exactly 11 after 9. If there are there any more questions that you would like to ask? Do I see? Yeah, I see another hand. So that will most probably be the last question of the discussion today. And
thank you again. One more question. Maybe it will be kind of heretic, but I will try. We have the Human Brain Project that can make a map of a specific brain of a specific human, right? So can this model, this map of a human brain be used to make a simulation and artificial intelligence that would simulate this person's brain activity? If you know what I mean. I can say it, explain it in more details if you need. Maybe I don't know if you understand what I mean.
So I understand that you are referring to simulation, which was very much in the focus of the Human Brain Project, in particular in the beginning years. I would like to say that simulation is always an approach to simulate something very specific. It's always purpose-driven. And it was not the purpose to simulate abilities of a person. It was a purpose to simulate the activity of nerve cells under certain conditions or to simulate a certain learning process. And when you look to simulation in, following these lines, then it becomes extremely helpful and powerful experiments. And during the past years, we have developed in the Human Brain Project different simulation approaches, starting from molecular simulation to cellular simulation with very high detail of single neurons, to larger network simulation and to simulations of the whole brain, but not with so much detail. And now bringing these different approaches together, we can ask more and more specific and also more complex questions, but it's always purpose-driven and not the simulation of the brain per se in itself.
But could it be possible in the future to make a simulated artificial intelligence simulation based on this brain model?
A brain model is always a mathematical model or a data model, and so to say, a simulation is a process that is based on this. So the more you know about the mathematics behind or about the data, the better you can simulate.
Okay, so there will be no cloning of a person based on these models.
well, it all depends on the accuracy, right? You can come up with models right now that will model people, right? I can predict, for example, how my kids will behave in certain circumstances, even without mathematical modeling, right? So that's already some kind of approximation. The more we know about the brain, the more precise we are. Still, it's a model. And if you are asking a question whether we will be able to replicate brain one-to-one, like clone it one-to-one, I mean, first of all, why would we want to do it? Why would we want to direct our resources into cloning, into making exactly identical copy of a human? Like, what would be the benefit? There is a certain level of approximation that is useful for us, to maybe to describe patterns in human behavior, maybe to, when we have those patterns, we can look for abnormalities related to aging, to disease, but pushing it beyond certain level would never be, I mean, I don't think it would be useful or I don't know if it would be achievable, considering the number of neurons and the synapses. Creating a one-to-one replica of human nervous system would not be, is not feasible, but also it's not really necessary. But at each level of our understanding of the brain, we do have a simulation that shows us how the brain works, right? And we will push it farther and farther until we find it, let's say, useful or detailed enough, right?
It could be beneficial, this simulation of a specific brain when it comes to because okay, I am a member of a transhumanism society, and in the transhumanism society we discuss such problems like digital
you.
In digital immortality. And this is what I was thinking about. Is it possible in that way?
Yeah, that's, I don't think we can answer that question at this point. I don't think whether you can upload your, because, so the question is, what does it mean that, what makes you you, right? So we tend to think that what makes a person are his personality traits, right? Impulsivity, I don't know, sociability, but it's not always, I don't think it is the case, you know? I think the memories and experience is what makes us the human, right? Or to a large extent. And I think it might be possible to generate a simulation of one's personal memories and upload it onto a computer. Will it be useful? Will it make that person happy? Will it fulfill your dreams as transhumanists? I'm not sure about it. So actually, the fact that we are mortal is also a part of being of the experience of being a human. So you have to consider that as well. Well, are we allowed to take one more question? We're not. Oh, please. Just one more.
Thank you very much. I just wanted to ask about AI tools, because I heard a lot of discussions considering the topic, that they are making the work faster, easier, and more efficient. And have you tried, or are there any AI tools that are being used in neuroscience, computational science, neuroscience? And are you perhaps using them in your project?
So we are using different AI tools, deep learning, machine learning. I mean, there's a whole variation. They are particularly powerful for all kind of image analysis and image classification. So here we benefit, of course, a lot. I see that from studying the networks, for example, axonal networks, we may learn more about how networks are being structured in order to fulfill a certain function and a certain task. So I see that this is a bilateral relationship between we are using AI, but we are also informing AI in the future. There are still fundamental differences between artificial neural networks and natural neural networks, but to analyze where precisely are the differences will help us a lot in both sciences, I would say.
Well, in my opinion, the AI tools will really give a big push to neuroscience, because they allow analyzing much bigger data sets in a very fast and efficient manner. So we don't have to sit and look for even identifying neurons, identifying neural classes, identifying artifacts, anything, you know, with the help of properly assisted artificial intelligence becomes much more efficient and much more streamlined these days.
But on the other hand, causal relationships are not yet there. Yeah? So why is something developing like this? This is
True.
one of the questions where we cannot yet use AI.
Yeah, you can see it even with the artificial intelligence that we interact with now, that it's not as good as, it's good at generating generic answers, but it's not as creative as humans still, I would claim.
It also needs a lot of input data. The current AI needs to be fed millions of data
That's
points.
a big
And
problem
this is
for
what
human brain neuroscience. We do not have so many input data, but so to say, single-shot learning, yeah, or unclassified, unsupervised learning. So these are approaches that are interesting, of course. Yeah.
Okay, let me just check all the input
I
sources.
think the
to my brain.
organizers are
Whether we
telling
have
us
any more
that
questions,
it's time to wrap up.
So let's wrap up this discussion and thank our guest, Katrin Amunds, the Scientific Director of Human Brain Project, our guest, Marcin Szwed from Jagiellonian University, and me, Rafał Czajkowski. I would like to thank you for participation and for active participation and for making it possible and for great 45 minutes.